Yeah, thank you, Mark, for the nice introduction. And oh man, is it good to be here finally. I could also say I am deeply shaken after Monday's approach to the airport. Yeah, so I'm not Frank, I'm going to talk about the genome anyway, but also he's going to be the good cop, I will need that. Maybe I'm a bit afraid I have too many slides, but don't worry, I'm going to deviate from them anyway very soon. So, yeah, why am I working on this? I want to make clear my motivation and maybe my upbringing. So I'm a bioinformatician by design, and I call myself Salmon Louse bioinformatician, which is maybe a rare species, especially for those that are this by their upbringing, in a sense. So sometimes I talk to other bioinformaticians and say, hey, stupid. I mean, it's like, why am I working with this animal, right? I mean, for them, genomes are just arrows on lines, right? And genomes have some sizes and letters. But, I mean, for me, this animal is very interesting because it is so well adapted to, well, its niche, and I think it has so many interesting features that we can study further. So I believe that every sort of genome contains some sort of gold nugget in itself, something you maybe could only find in this genome and in this animal, in this system, and maybe not anywhere else. Or maybe you could, but very difficult. So, speaking of gold nuggets, I think there's maybe somebody in the audience who rather want to give me a silver bullet to kill this animal, right? But I'm not much interested in that. In fact, I don't want to kill the louse, I want to study it. Do you find that offensive, by the way? No. Okay, I didn't think so. So, yeah, what's the genome, in fact? I mean, the genome is the blueprint for all proteins in the cell. We all know that. And also the non-coding RNA species. It is highly plastic and dynamic, right? The view has shifted much from the static set of letters, like you can print out as a book or something. And then it's highly variable. It's changing all the time in all the cells. And only a tiny fraction of it is coding for the polypeptide. So then parasitism, because we're dealing with a parasite, and that's what I'm going to talk about, right? How, in principle, how the parasite or parasitism shapes the genome. So parasitism is a form of symbiosis. Yes, it is, in the strict sense, yes. It has some influences on ecology, food webs, and the like. And yeah, there are lots of parasites, right? Each organism is either a parasite or has one, at least, or many. So how does this affect the genome? And there was a very influential, I would say, review by Poulin and Randawa about this evolution of parasitism along convergent lines from ecology to genomics. And yeah, they study how parasitism could shape genome architecture. And here they say, nevertheless, as more parasite genomes become available, we may be able to detect clear trends in the evolution of parasitic genome architectures, representing true convergent adaptive peaks, the genomic equivalence of the phenotypic strategies used by all parasites. So brace yourself and buckle up, so time is nigh, right? So because we have the salmon louse genome, and we have, this is the Atlantic salmon louse genome, it was just published, and then even more recently, the Pacific salmon louse genome by Yoshi et al. So we have these assemblies, so we need to take a step back, right? What do we do with it? Oh, we need to take another step back, and I hope you're not asking this question, why did it take so long? So, because I'm going to answer it. So, obtaining the L. salmonis genome, the timeline was in principle planned and started in before 2009. And yeah, it was first how to obtain funding. We initiated the sequencing project. The sequencing was completed then someday, and in 2012 we built the SLC, and I established Lice base, and then finally in 2021 the genome was published. And of course this is a long time, but everybody who has tried to sequence and publish a genome, especially in times of high expectations, where you almost should get a Nature publication out of it, they will see that this is more like a process in extreme group dynamics than anything else. So you need a lot of team building activities to finally get through. And then it's better to have like a small core team of 3-4 people who do most of this stuff. So what did we sequence for this genome? Right? They meticulously inbred lice strains over 27 generations to reduce contamination and heterozygosity, because one animal obviously is not enough, or at least not at that time. And then we also had samples from all developmental stages from RNA-seq. And then the genome was sequenced and assembled. I'm talking about the Atlantic genome now, right? I'm not talking about other people's genomes. So and then we used a hybrid strategy with inbred lice. 454 sequencing was actually like top-notch. Illumina paired end sequencing, and we achieved 181-fold coverage, which is not bad, in fact. And then a lot was tried to assemble the genome, and then we ended up with a final assembly strategy with initial context made by the Nubler assembler this time, also top-notch for 454, and then correction of homopolymers by Illumina sequencing, scaffolding, and so on. And that was the best compromise of N50 and linkage breaks, mostly, yeah, and completeness. And then we also did the Busco analysis, of course, mapping of DNA and RNA sequencing, and so on. So then the genome was predicted. We had finally some funding to get Ensemble Metazoa do it. And we had some ESTs and RNAs training data. Maker2 was used, Interproscan, and you name it, Ensemble Compara. Then for the paper, we had to use AutoFinder again because the reviewers demanded to use other organisms than were in Ensemble. And yeah, like it always is. And then Busco was used to assess single autolog and completeness, right? So when we compare this, like it's like 10-year-old genome in the sense it's sort of from the time of the steam engine compared to the electric cars of now. But I'm very happy it's still not that bad. So, I mean, we have, right? Here's our L-salatl, and this is the newly submitted RefSec assembly of the Pacific version. And of course, you see we have like on the scaffold level assemblies, and they have chromosome. But we have a very high depth, but well, way too many scaffolds, of course. The N50 they have is much higher. That's what you can achieve with current technology, right? PacBio and stuff. Or Oxford Nanopore. The GC content is reassuringly the same. Repeat content also, I will go into that later, and also the coding genes, even though people tend to brag about the number of genes, so in every paper they have more and more genes, because it's good to add more genes, but when you look at the coding genes, we were just missing, well, a tiny fraction, I would say. And then when you do Busco, in fact, you see that our genome is pretty good. In fact, it is the most complete L-Salmonis assembly as of today. In terms of Busco, of course. Of course, I mean, our context, right? Our scaffolds, they like this. And the Pacific biosequence, or the other ones, so it's almost impossible to compare. But I think most of this variation, or in the length, or what we're missing is in repeats. Okay? So we did a repeat analysis with repeat masker, and all the L-Salmonis assemblies assured us that we have the highest repeat content of any crustacean genome in the Atlantic salmon louse, above 60%. So for comparison, the Atlantic salmon also has about this. The locust, Locusta migratoria, has the largest sequenced arthropod genome, I think, at the moment, has also 60%. And the axolotl, the largest animal genome sequence, has about 65%. So we're quite there. So I compared this to other assemblies. Here's the L-Salmonis assemblies and the repeat composition with retroelements, DNA transposons, unclassified. Most is unclassified, unfortunately, because they need to be better studied, I think. But there are a lot of autonomous genetic elements in there, right? Retrotransposons and DNA transposons when you compare to the other crustaceans here. And here's Rogacressii, the genome, and there, yeah, it's almost the same with 57%. So this is great to find out, but what is it good for? I mean, it's just repeats, right? It's the formerly called so-called junk DNA. I shouldn't say this loud, but anyway. So may this affect something? And in fact, there are many papers now stating that repeat content and especially transposable element content affect genome evolution, genome plasticity, maybe the adaptability of the organism. Also, there is like solid evidence for horizontal transposon transfer between different species, including fish and parasite. But of course, we haven't had time to establish whether this is the case in the salmon. So if anybody wants to pick that up, I would be glad or to work with them. So, and it gets even better. So now we don't even know how big the genome is. We know how big the assembly is, right? The assembly is, well, 600, 700 megabases. Okay, but seemingly it may be much bigger. Also, when you have these measurements, this is a paper I really would have loved to see here by Wingard, and where also Rasmus and Hjeti were involved. Yeah, I mean, they're just stating, yeah, we don't know the genome size right now. And I mean, we knew from before that assembly sizes do not really constitute the genome size. But we seemingly missing a lot. We're not quite sure how much we're missing. Even the genome size may be variable. We need to find that out. So, but now, this is the one parasite genome. What is special about parasite genomes, right? And one reviewer in the process made us really formalize this with p-values. I initially didn't want to go into that minefield, but seemingly nobody had done that before because they always claim parasitism leads to loss of function, right? But nobody had shown this with a p-value. So we could show it, and we showed that host-associated genomes have significantly less genes and higher, significantly smaller predicted proteomes than genomes of free-living organisms. So smaller proteomes and less genes. We define this proteome information content, just take the proteome, remove all duplicated isoforms, and then count. I included now 32 species, 16 parasites, 16 host-associated, and 16 parasites. And this is an extended set with respect to the genome paper. In fact, I tried a lot to sort of massage the data to get rid of this effect because I was a bit uncomfortable with it, but I didn't succeed. The more I add, in fact, the more pronounced it gets. So it doesn't really work. So it's significant, and you can't really get it to go away. So what you can see is here on the left, there's a graph with the number of sequences. So the number of sequences is significantly different, right? And here I have all the free, and here the host associated on the right side. And then also I separate by lifestyle free, like the real true parasites, parasitoid, plant, pest, social, and so on. So put everybody that has this host in here, and the free ones here, also the social ones, like bees, for example. And we see also this for the total length of the proteome. And especially you see that the true core parasites, especially, they have very much smaller genomes. This effect is also persistent and reproducible for all the taxonomic groups we studied. So whenever we have a group where there's parasites or host-associated species and free-living ones, then always the parasite has the smallest proteome. Great. So what's missing, right? Well, there's a lot of incomplete compacted pathways and gene sets. Incomplete heme pathway, I'm going to go into detail, lack of peroxisomes, few genes for stress response and detoxification, small set of chemoreceptors. If you want to know all, you just need to look up in the paper. So this is the hypothesis, like loss of function is the adaptation to parasitic lifestyle. So what's expanded? Not that much. There are only a few expanded domains and gene families, SHK domains, these FN2 domains, the fibronectin type 2 domains. that were mostly known from vertebrates from before, astacenes, and we got void genes. So you can have a look at Marlene's poster if you want to know more about this. Yeah, and then I remember once I talked to a colleague, and she was like, why do you work with this organism? Why don't you work in biomedical research? You could do so much good. I was like, okay, maybe we can do that too. So here, what we solved here is the mystery of intestinal heme absorption. Heme is an essential cofactor, iron-containing, and in fact, in the human, it is not quite known how this works in the intestine. There have been some papers, but a lot of discussion. Something turned out to be not correct, but we found this in the louse. So the louse is a natural heme auxotroph, seemingly. So here are the heme synthesis and degradation pathways in KEG, right, from starting here. And we only have this one left on this pathway. And of course, louse and salmon, we have to treat them as a system. So together, of course, right, the pathway is complete, obviously. So it is quite logical that the louse must retrieve heme from the host. And this is the protein that does it most likely. A scavenger receptor we found highly expressed in the intestine. We call that Eysturskarð for heme scavenger receptor. Yeah. A receptor CD36-like protein is the mediator of intestinal heme absorption. Complicated title. So heme is also toxic. Talking about detoxification, we checked different gene families and domain families for relation to detoxification. So there are several ones. Of course, this is a bit subjective what you choose, but we started here with the cytochrome P450 that were already studied by Humble, the ABC transporter-like that were studied by Carmona Atonanthas, glutathione S transferases that were not studied before. They are all compacted. So it's not like this, like we thought maybe. This organism is very good in adapting, right, to a toxic environment, all the toxins we put at it. But this doesn't mean these genes or gene families must be duplicated in any sense. That was maybe, in fact, a bit naive to think about this. So the only thing that's there is the vault again. Here that may be related to stress response. It's unknown what it does. The heme peroxidase superfamily also was studied by Overgaard. Should mention this. Also reduced. So in principle, when you sum it up, in fact, in total, you have like a lack of 200 detoxification or stress-related genes in comparison to the maximum what you get in this group. So, in conclusion, sea lice research has received a significant push with the availability of the genomes. I think we couldn't have done a lot of stuff also presented today without the genome, I think. We found loss of function is the hallmark of parasitism. What does that mean for sea lice control? I think it's a bit gloomy in a sense, because, I mean, there are less targets, and maybe the most exposed targets are already away by evolution. Also, there's the high repeat content that makes the genome very flexible to, and gives plasticity to restructure everything in case it is needed. So, at least it doesn't increase the optimism, sort of, these features. But we will see. In limited aspects, like heme uptake, intestinal heme absorption, we could push or pull ahead of model organisms and biomedical research, because that we could do experiments we can't do in humans, really, right? Maybe in human cells, but anyway. Driving force here is open data. So I have the policy making everything open, mostly, fair share principles of data, and of course the right people to look at them. And now, for closing, maybe I can show you this little video of the intestinal heme receptor. Will it play now? Yeah, so just everything starts, of course, with genomic sequence, and then it gets transcribed and translated into the protein sequence, but this is just linear, and then, of course, it needs to fold correctly into the three-dimensional structure of the heme receptor, in this case, right? And finally, this thing sits in the membrane of the intestinal enterocytes. And then maybe there's a heme molecule coming. Now, this is a bit of science fiction, so I'm not quite sure if it really works like this, right? So it docks maybe here. There are several docking positions for the heme molecule, and then it migrates through this into the channel. Of course, it was a bit, well, it's a bit imagination in this case. Just the channel is there, and it definitely doesn't say click when it attaches to the docking position. So, to wrap up, thank you all. Acknowledge a lot of people, also for the heme receptor, for the genome, right? I should acknowledge my funding, Elixir Norway, Forskningsrådet, BioStars, also to spend my time and get good ideas. Yeah, and there we are. Thank you for your attention.